Hello, and welcome to Task 1 Biology with Theodore. Today, I'll be answering a few age-old questions all relating to DNA, its structure, how it is replicated, as well as how it turns into proteins. This is DNA. Wait, actually, this is DNA. As we can see, it consists of two strands that twist together to form a double helix. The rungs of the ladder-like structure are made from pairs of bases called nucleotides.
But firstly, we need to give a little backstory of DNA and its purpose. DNA is found in the nucleus of cells as chromosomes, where DNA is tightly coiled around proteins called histones. Within the DNA are sections called genes, which hold the instructions for making proteins.
Let's zoom in. Each nucleotide has three main parts: a phosphate, a sugar, and a nitrogenous base. Where the phosphate links each nucleotide, the sugar helps as a backbone, and the base carries the code. Here we have two half strands going anti-parallel. We can tell this by noticing the sugar. What is important is that the way the carbons are numbered in that sugar, where the 5' is up by the phosphate and the 3' is down by the flat part of the sugar. This gives us a direction to read the bases. We didn't always know what DNA looked like. Thanks to Rosalind Franklin's crystallography photo 51, scientists could determine its structure. Using Franklin's data, James Watson and Francis Crick built the first accurate DNA model in 1953. Looking back to our model, we can see four kinds of nitrogenous bases. They are adenine, thymine, as well as guanine and cytosine. They are complementary paired, meaning A always goes to T and G to C. They are held together by hydrogen bonds, double bonded between adenine and thymine, and triple bonded between cytosine and guanine. Before a cell can divide, it needs to make an exact copy of its DNA through DNA replication. First, an enzyme called helicase comes and unzips the DNA by breaking the hydrogen bonds between the base pairs, creating a replication fork. Then, an enzyme called primase adds a short RNA primer to one strand, allowing a starting point for DNA polymerase to bind and start adding nucleotides. To note, RNA is slightly different from DNA because it uses uracil instead of thymine and contains ribose sugar instead of deoxyribose. Now, DNA polymerase moves along the original strand, adding complementary nucleotides, going in the 5' to 3' direction, adding in one continuous strand. This is called the leading strand. On the other side, the same process is repeated with primase and polymerase, but remember how polymerase only goes in the 5' to 3' direction. Well, polymerase and primase must create a lot of short segments to fully replicate this strand. These short segments are called Okazaki fragments. This completes the lagging strand. Another enzyme called exonuclease removes all RNA primers from both strands, while another DNA polymerase fills in the gaps. What is important to remember is that helicase, primase, and polymerase are all working simultaneously, splitting old DNA and creating new. Finally, ligase seals the DNA fragments created by DNA polymerase together, forming continuous double strands without any gaps. In the end, we're left with two identical DNA molecules. Now that the DNA has been replicated, the cell can use this information to make proteins. The first step is called transcription, occurring in the nucleus. The enzyme RNA polymerase, different from DNA polymerase, attaches itself to the DNA, knowing where to start because it recognizes a specific DNA sequence called a promoter. Only one strand of DNA is used as the template, usually the one which goes 3' to 5', as polymerase can only write in 5' to 3'. RNA polymerase moves along the DNA and builds a complementary strand of messenger RNA, or mRNA, again using U in place of T. This process continues till RNA polymerase reaches a terminator sequence and detaches from the DNA. Once completed, a strand of pre-mRNA is formed containing non-coding sections called introns and coding sections called exons. Introns are cut out while the exons are joined together, producing mature mRNA, as well as adding a 5' cap and a 3' tail, increasing likelihood of mRNA reaching its destination effectively. The mature mRNA leaves the nucleus and travels to the cytoplasm to find a ribosome. Before we keep going, rRNA or ribosomal RNA is what makes up the ribosome, which is the structure that builds proteins. Now that the mRNA has reached the ribosomes, it's time for translation to occur, where the ribosome reads the mRNA code and starts building a protein. The ribosome slides along the mRNA in lots of 3 called codons, looking for the start codon, usually AUG, which tells the ribosome to begin. tRNA carries a specific amino acid and has an anticodon that matches that of the mRNA. Using the code read by the ribosomes, tRNA supplies rRNA with the materials to build a protein. When tRNA pairs with the mRNA codon, the ribosome links its amino acid to the growing chain by forming peptide bonds, building a polypeptide chain. The ribosomes keep moving along, reading in lots of 3, adding their corresponding amino acid and leaving. After a while, a long polypeptide chain is formed. Finally, the ribosome hits a stop codon, signaling it to, stop. The polypeptide chain is finally complete and all components dissociate, allowing the new polypeptide to fold into a 3D shape and become a functional protein. And thank you. Thanks for watching my video, teaching me biology, and being a really cool teacher. I just want to say thank you for everything you've given me, and I can't wait to see what else we can do with Miss Ruse. Goodbye.